Hello, my lovely kids. I hope you all are doing well. So, today we are going to start our revision of our very first chapter, that is Cell, the Unit of Life. This is a very important chapter because of this chapter you will get to know the basics of other chapters, right? So this chapter will connect you to the other chapters. So if you have basically skipped any of the lecture or any of the topics, so don't worry, we will cover everything from this chapter. We will revise everything. So without any delay, let's start with our revision. So we will first start basically with the introduction, then we will discuss about cell theory, then we will discuss about prokaryotic cell, and then eukaryotic cell. Okay, so first we are going to discuss about the introduction. Basically, what is a cell? We all know that the cell is the structural and functional unit of living organisms, right? We all know that cell is basically the structural and functional unit of all living organism. It means that it determines the structure of the organism, and every function that our body perform is because of cell. We are able to walk, we are able to talk, we are able to think, we are able to process anything, we are able to walk, we are able to write. Everything is because of cell, right? So cell makes the structure of organism, and cell performs the various function inside our body, because our body is made up of cell, right? We have millions and trillions of cell in our body. Let's discuss about the discovery of cell, that how this cell was discovered. So cell, basically, first of all, the name comes as Robert Hooke. The first name is Robert Hooke. Robert Hooke basically discovered the dead cell for the first time. He discovered dead cell for the first time. And what about the living cell? Who discovered the living cell for the first time? Anton von Leeuwenhoek. Anton von A.V. Leeuwenhoek, or Anton von Leeuwenhoek, he discovered living cell for the first time. Living cell for the first time. And the third name that we need to remember is of Robert Brown. Robert Brown is basically responsible for the discovery of nucleus. It is Robert Brown basically is responsible for the discovery of nucleus. What is this nucleus? We have already discussed a very brief structure of cell. I'll draw it here. There are basically three main parts of the cell here. This is what we call as cell membrane. This is what we call as cytoplasm, right? And this is what we call as a nucleus. Inside nucleus, you will find the basically DNA, right? You will find the DNA that controls all the functions of cell. That's why nucleus is known as control house of the cell. And we know that the cytoplasm and nucleus together, they are known as protoplasm. What is a protoplasm? Cytoplasm plus nucleus together, it is known as protoplasm, right? So instead of saying cytoplasm plus nucleus, we can say protoplasm, right? It is a combined term for cytoplasm and nucleus, right? So I hope everything is clear till here. This is just the introduction. So after this discovery of cell and various structure of cell, right, meanwhile several theory were coming up. regarding the cell. So there were two scientists, Skeldon and Schwann. There were two scientists, Skeldon and Schwann. Skeldon basically was German botanist. He was fond of plant, and Schwann was British zoologist. He was fond of what? Animal. So Skeldon studied plants, various plants, and Schwann studied various animal. Both came to the same conclusion. They formulated cell theory. They formulated cell theory. They gave the cell theory. They both came to the same conclusion that every organism on this earth is made up of various kind of cell, right? Like in plant has parenchyma, collenchyma. I hope you remember this. You have, I think, already studied this in your 9th standard. Parenchyma, collenchyma, sclerenchyma, xylem, phloem. So plants are basically made up of different kind of cell and tissue, and similarly animal is also made up of different kind of cell and tissue. So both of them studied plant and animal, but and also came to the same conclusion. But they both failed to tell us that how every new cell arises, right? How a new cell come. So this was told by Rudolf Virchow. Rudolf Virchow basically told us that new cell arise from pre-existing cell. So Rudolf Virchow said that every new cell arises from their parent cell, which we called as a cell division. This is the parent cell. These are daughter cell. We call it cell division. Right? So Rudolf Virchow told us that every new cell arises from pre-existing cell. Right? Now we know it is through cell division. Right? So you have to remember these three names, Skeldon and Schwann, both gave the cell theory, but Rudolf Virchow modified cell theory. So if the question comes that who modified the cell theory, it was Rudolf Virchow. Okay, so these were the three names that you need to remember, and they are very important. Next comes about the cell size and cell shape, right? Do every cell has basically the same shape, same size, right? Or do they differ in size and shape? So yes, there's a lots of variation in cell size and cell shape, right? Let's talk about cell size. Let's talk about the cell size. But cell size, like smallest cell we know is mycoplasma, which is a bacteria. Mycoplasma is basically the bacteria. It is also known as PPLO. It is known as PPLO. So in theory, it has been written that mycoplasma is approximately 0.3 micrometer. And in diagram, it has been written that PPLO is 0.1 micrometer. So don't get confused. Just remember both the data. If mycoplasma comes, you have to choose 0.3 micrometer, and if PPLO comes, you have to choose 0.1 micrometer, right? There is no need to get confused here. Both are the same thing, mycoplasma and PPLO. But the data that has been given in NCERT is different. You need to remember both. Then the largest cell, you know, ostrich egg cell, right? You know the longest cell, nerve cell. And what about the virus? Virus does not, basically we don't include virus in a cell because virus is not a cell, right? So we are not going to write it here. Next we will talk about cell shape. Cell shape also varies. Cell don't have a specific shape, which means every cell is not same in with regards to shape. Like for example, your nerve cell is very long, as you know, and branched. You must have studied in this zoology. Nerve cell is very long. Red blood cell, it is basically oval and biconcave. So basically RBC shape is like vada or donuts without the hole in between. So the shape of RBC is that only. White blood cell, if we talk about white blood cell, they don't have a specific shape. So we call it amoeboid shape, or we call it irregular shape, right? So these are the data that you need to remember, that yes, cells are not same in size and cell are not same in shape, right? So every cell is differ in shape and size from other. So this is the data that is very important for you to remember. This is a very small thing, but yes, you need to remember this. I hope everything is clear till here, because this is just an introduction for you guys. So now the types of cell on the basis of complexity, How complex is the cell? So on the basis of this, there are two type of cell. Beta. Right? There are two type of cell. First is prokaryotic cell, and second is eukaryotic cell. So on the basis of complexity, there are two types of cell, prokaryotic and eukaryotic. What is the difference between? Pro means primitive, karyotic means carrion, which is nucleus. They have primitive nucleus, whereas U means true and karyotic come from carrion. Carrion means nucleus. They have true nucleus. Primitive nucleus, which means old type of nucleus, and true nucleus. What is an old type of nucleus? In which there is no membrane around the genetic material. Nuclear envelope is absent. There is no covering around the genetic material. Whereas here, nuclear envelope is present. There is covering around the genetic material in eukaryotic cell. Next is right? Next is that they don't have membrane bound organelle. So membrane covered organelles are absent. There is no organelle in prokaryotic cell which is covered by membrane. Whereas membrane bound, bound means covered, membrane covered organelle are present. For example, ER, Golgi body, etc. So yes, membrane covered organelle are present in eukaryotic cell, but membrane bound organelle are absent in prokaryotic cell. Membraneless organelle like ribosome is present. There is one organelle in prokaryotic cell, and that organelle is ribosome. That organelle is ribosome. And there are two membraneless organelle without membrane, like ribosome and centrosome. Are present, right? So prokaryotic cell has only membrane-less organelle, whereas eukaryotic cell has both membrane-bound and membrane-less organelle. So both type of organelle are present in prokaryotic cell. Example of prokaryotic cell is only bacteria. There is only one example of prokaryotic cell, which is bacteria. So all bacteria on this earth, right, has prokaryotic cell. Whereas example of eukaryotic cell is fungi, plant, animal, protist, right? So any other organism other than bacteria are having eukaryotic cell. So only bacteria are having prokaryotic cell, and other organism like fungi, plant, animal, and protist. What are protist? Amoeba, paramecium, euglena, all are protist. They have eukaryotic cell. So these are the type of cell that we are going to study in detail. So first we will take with prokaryotic cell. Right? So the only example is bacteria. So yes, we are going to make a diagram of a typical bacterial cell. This is the plasma membrane. This is the cell wall. And this is the glycocalyx. There are 3 layers. In the bacterial cell envelope. I'm going to label and tell you the function of all the basically structure of bacterial cell here only. So we will start making the diagram here. This is the flagella with the help of which the bacterial cell moves. It makes the bacterial cell motile. And these are the tube-like structure that helps the bacteria in mating. And these are the hair-like structure that helps the bacteria in attachment. Okay, and there is some structures. There are some structures. And this is the genetic material. This is the ribosome. Many ribosomes. I'm going to tell you what are these. And these are the reserve food material. Okay. So let's start labeling the diagram here. This is a typical bacterial cell. Beta, this is the outermost covering. We call it glycocalyx. Right? So this is the outermost covering. Glycocalyx may be loose sheath. When it is loose sheath, it is known as slime layer. Right? Loose means very thin layer. When it is very thick layer, it is known as capsule. It is known as capsule. Glycocalyx basically gives the very sticky nature to the cell. It makes the cell very, very sticky, so it will be easier for cell to attach to any surface. Right? Second is, this thick layer is known as cell wall. Right? The cell wall of bacteria is made up of peptidoglycan. Is made up of peptidoglycan. We also call it murin. Peptido means peptide, means amino acid. Glycan means sugar. So cell wall of bacteria is made up of amino acid and sugar. I'm again telling, peptide means amino acid, a chain of amino acid, and glycan means sugar. So the cell wall of bacteria is made up of amino acid and sugar. Right? Third layer is plasma membrane. We are going to study the structure of plasma membrane in eukaryotic cells, so don't worry. Here we are not going into the details. So these three layers, glycocalyx, cell wall and plasma membrane, together they are known as cell envelope. Together they are known as cell envelope. So there are three layers in the covering of cell, bacteria cell: glycocalyx, cell wall and plasma membrane. Right? Now we will see other structures one by one. Beta, this is flagella. Right? Flagella is made up of protein, flagellin. It is made up of flagellin protein. Right? And it may or may not present. It is not necessary that it is present. So it may or may not present. And it provides motility. It provides motility, sorry. It's provide motility, which means the bacterial cell can move with the help of flagella. So here the flagella is made up of flagellin protein. It may or may not present, and it provide motility. And flagella has three parts: base, hook and filament. So this is the base, this is hook, and this is the filament. It is the longest part, right? So the longest part is filament. This is the base, this is the hook, and this is the filament, which is the longest part, right? So this is all about flagella. Let's talk about these hair-like structures. These are known as fimbriae. These are known as fimbriae. These hair-like structures are known as fimbriae. They help in attachment of bacterial cell to any surface. So they help the bacteria to get attached to any surface. Bacterial cell to get attached to surface, okay, to any surface. So fimbriae, basically they are small hair-like structures that help in the attachment of bacterial cell. Now these, what are these? These are pili. They help in mating. Mating means transfer of a part of DNA from one bacteria to another. So pili helps in mating, right? What is the meaning of mating here? The transfer of a small fragment of DNA from one cell to another. This is chromatophores. What are these? These are the extension of plasma membrane. As you can see, the plasma membrane gets folded inside towards the cytoplasm. So chromatophore are basically extension of plasma membrane, right? What is the function they do? They help in photosynthesis. Because they contains pigment. Pigments help in photosynthesis. Pigments help in capturing the light and performing photosynthesis. So as these structures contain pigments, they help in photosynthesis. Next is this structure, beta. This structure is known as mesosomes. This structure is known as mesosome. Mesosome, basically it is also an extension of plasma membrane. It is also an extension of plasma membrane. It helps in respiration. Chromatophores were helping in photosynthesis. Mesosome helps in respiration, also in cell wall synthesis. They also give material for cell wall synthesis in new daughter cell. They also help in DNA replication, which means making copy of DNA. They also help in secreting out some chemical, right? Secretion means to give out any chemical, right? So they also help in the secretion of some chemical. So these are some of the structure that we have studied till here. There are 3 things that have been left. So I will just label them. These are genetic material. This is genetic material. Beta, this is polysome, right? This is polysome. So about genetic material and polysome I am going to tell you in the next slide. And this is bache inclusion body. So there are 3 things that we are going to discuss in next slide. That is genetic material, polysome and inclusion body. I hope everything is clear till here. Anyone? So let's start with genetic material. Right? Genetic material is without membrane or envelope. There is no envelope or covering around the genetic material, right? And the DNA with protein is present in the region of cytoplasm, and that region is known as nucleoid. What do we call that area where genetic material is present with protein? That area is known as nucleoid. What kind of DNA is present? DNA is double-stranded circular DNA. There are two strands. And both the ends are sealed. They are two strands. And both ends are sealed. So which means the DNA is double-stranded circular DNA. So what kind of genetic material is present in bacteria? It is double-stranded circular DNA. Then we will talk about polysome. What are polysome, beta? So what happens during protein synthesis? There is a chain of messenger RNA. This is messenger RNA, also known as mRNA. And many ribosomes get attached to it. These are ribosomes. Ribosomes are made up of rRNA and protein. Remember this, ribosome is made up of rRNA and protein. So there are many ribosomes. Get attached in a single RNA, get attached in a single mRNA. This chain is known as polysome or polyribosome. This chain is known as polyribosome. What kind of ribosome is present in prokaryotic cell? 70S. We have already discussed about this. In prokaryotic, there are two types of ribosomes, 70S and 80S, right? So what kind of ribosome is present in prokaryotic cell? It is 70S. Let's talk about inclusion body. Let's talk about inclusion body, beta. What are inclusion body? Any reserve food material. Any reserve food material is stored in the form of inclusion body. Example, glycogen, phosphate, cyanophycine granule, type of starch, and gas vacuole. So these are the food material that has been stored in bacteria, so they are also membraneless. Remember, they don't have any membrane. They are membraneless. So inclusion body is not covered by any membrane. So this was all about prokaryotic cell. Now we are going to study about eukaryotic cell. I hope everything is clear till here. About the genetic material, polysome. What is polysome? When many ribosomes get attached to a single mRNA, this is known as polysome. And inclusion body, any reserved food material is stored in the form of inclusion body. Let's go into the eukaryotic cell, bache. So I'm going to make a diagram of eukaryotic cell. Example is plant cell, animal cell, fungal cell, protist cell. So here I am going to make a diagram of a plant cell. Okay. This was the plasma membrane. This is cell wall. Okay. This is a vacuole that basically cover almost 90% of the plant cell. We can also modify it from here. So this is the vacuole that basically covers the 90% of the volume, and there's a nucleus here. Inside nucleus you will see the thread-like structure, which is the genetic material, and this is the nucleolus. And at the outer wall you will see rough endoplasmic reticulum. And here you will see the smooth endoplasmic reticulum. This is smooth endoplasmic reticulum, and this will be your Golgi body. Now I'm going to make a one diagram of here of, okay, this is mitochondria. This is chloroplast. And these are some microbodies. Sorry. So this is a typical plant cell. I just draw this diagram to make you understand. I didn't want to make any complex diagram here. So beta, this is the cell wall. Okay. And plant cell wall is composed of, this is a plant cell. Okay. This is a plant cell, and plant cell wall is composed of cellulose, hemicellulose, all are sugar, pectin, and proteins. So basically cell wall of plant is made up of sugar and protein. This is the plasma membrane. right? This is the plasma membrane. So plasma membrane is mostly composed of lipid and protein. But sugar, right, cholesterol are also present, right? But mostly in the plasma membrane, lipids and proteins are there. I'm going to make a detailed diagram of plasma membrane in the next slide. This is beta chloroplast. I'm going to make a diagram of a chloroplast here. Chloroplast has two membrane, right? Chloroplast has two membrane. And it has a disc shaped structure which is known as thylakoid. And the group of thylakoid is known as granum. Group of thylakoid is known as granum, and one granum is attached to another granum through stroma lamella, right? And this is known as stroma. Remember this, that chloroplast has double-stranded circular DNA just like bacterial cell. So there's a theory regarding this, that chloroplast basically derived from the bacterial cell. It has double-stranded circular DNA. I'm going to make it here. This is the double-stranded circular DNA. Okay. So this is the chloroplast, and chloroplast, as you know, helps in the photosynthesis. Now this is Golgi body. Golgi body helps in packaging. Material coming from endoplasmic reticulum. So Golgi body basically helps in the packaging of material that is coming from ER and sending it to the target. Right, and glycoprotein and glycolipid also prepared or synthesized here, and glycoprotein and glycolipid basically are used in plasma membrane. Glycoprotein and glycolipid main site is in Golgi body. This is beta smooth endoplasmic reticulum, also known as SCR. It helps in synthesis of lipid and sterols. Synthesis of lipid and sterols. So lipid and sterols basically are synthesized in smooth endoplasmic reticulum. This is vacuole, and the membrane of vacuole is known as tonoplast. Remember this. The membrane is known as tonoplast. You can see this membrane. This is known as tonoplast. This is known as microbody. They are single membrane bound organelle that helps in oxidation process. Example, peroxisomes, glyoxysomes, right? So microbodies, basically they are covered by single membrane and help in oxidation process. This is mitochondria. This is mitochondria. As you guys know that mitochondria is also composed of two membrane. One is irregular and one is regular. So these are two membrane. Right? Inner membrane has a folding known as cristae. The foldings are known as cristae, and mitochondria also has DNA. Also has DNA. Mitochondria also has single-stranded circular DNA, right? And mitochondria, we all know, helps in respiration. It helps in respiration, right? So here, in the next slide, we are going to study about in detail about plasma membrane and about nucleus, right? So this is basically functions of all the organelle, that which organelle is involved in which function. So in the next slide we are going to discuss about the plasma membrane and nucleus in detail. So I hope everything is clear till here. So let's discuss about plasma membrane. Plasma membrane has universal structure. It is similar in bacteria, prokaryotic cell and eukaryotic cell. It has universal structure, mainly composed of, as you know, lipid and protein. So its most accepted model was given by Singer and Nicholson. Remember these two names. So most accepted model for the structure of plasma membrane was given by Singer and Nicholson. So basically plasma membrane is made up of lipid bilayer. There are two types, two molecules or two layer of lipids. This is the integral protein. And this is the external protein. I'm going to make a cell diagram here. So beta, this is a cell, right? This is a cell, and I have basically drawn this part. Right? So this part has been drawn here, a small part of the plasma membrane. This is two layers of lipid. We call it lipid bilayer, right? This is head and this is tail. Head is hydrophilic, it loves water, whereas tail is hydrophobic. Head basically hates water. So that's why tail avoids the water by facing each other, and head is facing the water because there is water here and there is water here. There is water here and there is water here. So tail is avoiding the water by facing each other. Okay. Now these are the two types of protein. This is what we call as a intrinsic protein. So on the basis of how easy for us to extract them, there are two types of protein, intrinsic and extrinsic. Intrinsic is also known as integral protein, and these are extrinsic or peripheral protein. Extrinsic or peripheral protein. There are some cholesterol in between for stability. This is cholesterol. Okay, and there are some sugar which is being attached to the lipid, right? And there are some sugar that has been attached to protein. The sugar which is attached with the lipid is known as glycolipid, and the sugar which is attached to the protein is known as glycoprotein. And which is the major site where these things are prepared, glycoprotein and glycolipid? That is Golgi body. I have written it here, na? Golgi body. Glycoprotein and glycolipid are synthesized in Golgi body, and they are present in plasma membrane. So when the sugar is attached to the lipid, it is known as glycolipid, and when sugar is attached to the protein, it is known as glycoprotein. Right? So this is the structure of plasma membrane. You should know that plasma membrane basically mostly involved in transportation. Plasma membrane first of all has semi-fluid nature because of lipid. Because of lipid, they have semi-fluid nature. So this is about plasma membrane. I hope this is clear to all of you. Next, we are going to study about the nucleus. This is the nucleus, one membrane of nucleus. This is the outer membrane of nucleus. Outer nuclear membrane and inner nuclear membrane, and the space between the membrane is known as perinuclear space. Perinuclear space, the space between the membrane, and there is a genetic material, we call it chromatin material, and this is nucleolus. And this is the pore, we call it nuclear pore for the transportation of protein and RNA in both the direction. Transport of RNA and protein. So RNA and protein, the transportation in both the direction is due to nuclear pore. This is nucleolus. Nucleolus basically is involved in the synthesis of rRNA, also known as ribosomal RNA. And we know rRNA is present in where? rRNA is present where? In ribosome. Ribosome is made up of RNA, rRNA and protein. And where these rRNA are being synthesized? They are being synthesized in nucleolus, inside nucleus. Okay? And this is the chromatin material. This is the chromatin material, beta. Chromatin material is made up of DNA, histone proteins, a very famous histone protein that helps in the packaging of DNA, and what? RNA, small amount of RNA, and other non-histone proteins. Okay, so chromatin material is made up of DNA, histone protein, RNA, and other non-histone protein. During cell division, what happens? Condensation. And because of that condensation, you get this type of structure. We call it chromosome. There is a condensation of chromatin material occurs. It gets packed, tightly packed, and this structure is known as chromosome, right? The center of this part is known as centromere, and these are sister chromatids because they are exact copy of each other. We have discussed this in class. These are exact copy of each other, that's why we are calling it sister chromatids, and the middle part is known as centromere. The middle part is protected by or surrounded by a disc shaped structure. We call it kinetochore. So this is the central part, and this disc shaped structure is known as kinetochore. This disc shaped structure is known as kinetochore. So this is the central part. We call central part as centromere, and the centromere is surrounded by a disc shaped structure known as kinetochore. So during cell division, this chromatin material get condensed to form a very condensed material. We call it chromosome. What do we call it? Chromosome, right? So chromosome makes it very easy for cell to distribute the DNA among the daughter cell, right? Otherwise it is very difficult for cell to distribute these threads. So these threads make sure that during cell division they condensed and form a very thick structure known as chromosome. I hope this is clear to all of you. We are about to end this recap and solve some very interesting questions. Before that, I'm going to draw a flow chart here about the organelles. There are double membrane bound organelle. There is single membrane bound organelle, and there is membraneless organelle. Double membrane, the example is plastids. In plastids we have chromoplast, the colored plastid, the leucoplast, the plastid which is colorless, store food, and chloroplast. All are double membrane bound organelle. And second is mitochondria. So both are with two membranes. Single membrane bound organelle is endoplasmic reticulum, right? And there is SER, smooth endoplasmic reticulum, and rough endoplasmic reticulum. Okay, I am going to tell you here that rough endoplasmic reticulum is involved in protein synthesis. SER is involved in lipid synthesis. It involved in protein synthesis. Then Golgi body. Okay, then lysosomes. Lysosomes help in digestion. Lysosomes have digesting enzymes. or hydrolytic enzyme, and vacuole, right, and microbodies, all are single membrane bound organelle. And remember that beta, ER plus Golgi plus lysosome plus vacuole, together they are known as endomembrane system. Together they are known as endomembrane system. Now we'll talk about the membraneless organelle, which is ribosome. Right? Ribosome is two types, 70S and 80S. Where do we find the 70S ribosome? Mitochondria, chloroplast. In mitochondria and chloroplast we find the 70S ribosome. Where do we find 80S ribosome? In cytoplasm and with ER, endoplasmic reticulum. Next is centrosome. First was ribosome and second was centrosome. Remember that centrosome is absent in plant cell. It is only present in animal cell. Centrosome is only present in plant cell. Okay? I hope this is all clear to you guys. So I'm going to make a very brief diagram of centrosome in the next slide. Centrosome is basically the fluid material and two cylindrical structure which are 90 degree to each other. So we call it centrosphere or kinoplasm, and these are two centrioles. This structure is known as centrosome. Okay. Centriole has the main protein which we call as a microtubule, and which is made up of tubulin protein. We are not going into detail of that. Centriole, as you know, it has 9+0 arrangement of microtubule. There are 9 doublets of microtubule which is present in centriole. Microtubule is basically the cylindrical structure formed of tubulin protein. Okay, so microtubule is composed of tubulin protein and microtubule form centriole. So centriole has 9+0 arrangement of microtubule, and microtubule is made up of tubulin protein. What are the functions? Functions is that centriole forms spindle fiber during cell division. We have discussed the cell division chapter. Okay, they forms basal body for cilia and flagella. So they form the base for cilia and flagella. Here cilia and flagella has 9+2 arrangement. Of microtubule. There it was 9+0, but here It is 9 plus 2 arrangement. It is 9 plus 2 arrangement, right? Microtubule is a cylindrical structure made up of tubulin protein, right? Remember this, centrosome is membraneless, right? Centriole is also membraneless. It does not have any membrane. Whereas cilia and flagella are membrane bound structure. Whereas cilia and flagella are membrane bound structure. Cilia and flagella are membrane bound structure. This is where we end our discussion. I hope everything is clear to you guys. So whenever you see these kind of lecture, the revision lecture, please make sure that you have studied from the lectures and the actual lectures and notes. Only after that you will be able to get these concepts, right? So this is about centrosome and centriole. My question for you guys, there's a homework. Yes, of course there's a homework. There's a homework for you guys, that difference between flagella of prokaryotic cell and eukaryotic cell. You need to write the difference between the prokaryotic cell and eukaryotic cell. Okay? This is your homework. Write down the difference between prokaryotic and eukaryotic cell. The flagella. Now we will solve some questions. Which of the following is not an inclusion body? What are inclusion body? Reserved food material. Reserved food material. Glycogen, phosphate, synophysin, polysome. What are polysome? This is mRNA. When there are many ribosomes get attached to the mRNA, it is known as polyribosome. So answer here will be B. Polysome is not an inclusion body. Polysome is not an inclusion body, right? Remember this, that ribosome is known as protein factory because it helps in the protein synthesis. We call it protein factory because it provides the space where protein is being synthesized. So here polysome is not an inclusion. It's not an inclusion body. The size of pleuropneumonia-like organism PPLO, smallest bacteria, also known as mycoplasma. I have told you that if it has been asked PPLO, you will write 0.1. If they ask you mycoplasma, you will write 0.3 micrometer. 0.3 micrometer. Next is select the correct statement regarding the cell membrane. Okay, let's see. The fluid mosaic model of the cell membrane was proposed by Singer and Nicholson. Yes, this model was the most accepted one. Most accepted model. It was given by Singer and Nicholson. Yes. Sodium and potassium ions move across the cell membrane by passive transport? No. By active transport. Active transport means with the help of ATP. When there is a transport with the help of ATP, it is known as active transport. So sodium and potassium ions, they do not move across the cell membrane by passive transport, they move through active transport. So this is wrong. Okay, this is wrong. You have to select the correct. The first option will be there. Protein makes up the 60 to 70% of the cell membrane. No, approximately 52% and 40% lipid. 52% protein and 40% lipid. Lipids are arranged in bilayer with the polar head towards inner side. No, head towards outer side, like this. So heads are towards outside and the tail are facing each other. Tail facing each other. So B, C and D options are incorrect. Only one is correct. That fluid mosaic model, it is the most accepted model of plasma membrane given by Singer and Nicholson. And we need to find the correct statement. So option number A will be here. I hope this is clear to all of you. And the last question is, when the centromere is situated in the middle of two equal arms. So there are many types of chromosome. When it is in the middle, it is known as metacentric. When it is slightly away from the middle, where there are two arms, we call small arm as P and long arm as Q. This is known as submetacentric. When it is closer to one end, very close to one end, P arm and Q arm, this is known as acrocentric chromosome. And when it is to the end, at the end, there is no P arm here, this is known as telocentric. This is known as telocentric. So these are four types of chromosome on the basis of centromere position. So when it is in middle, it is known as metacentric. Submetacentric, slightly away from center. Telo means end, at the end. Acro, towards the end. So when the centromere is at the end, it is known as telocentric. When towards the end, it is known as acrocentric. When in the middle, it is known as metacentric. When away, slightly away from the middle, it is known as submetacentric. So here our answer, because it has been asked the middle, at the middle, we will select the fourth option, which is metacentric. I hope everything is clear to all of you. So I really hope that all the concept, right, whether you skipped or you attend the lecture, all have been revised in this lecture. And keep working hard. We will basically, you will have more lectures like this in the future of other chapters as well. So make sure before attending these lecture, you come prepared. Like 60-70% prepared. Okay, so do the homework that I have given, that differentiate between the flagella of prokaryotic cell and eukaryotic cell. I will see you in the next lecture. Till then, take care. Bye.